Hi, everyone. For those of you that don't know me, my name is Christy Polly. I am Dr. Coughlin's lab manager. We are going to get started in just a few minutes here. I just want to remind everyone that we are recording this, so we will post that to our YouTube channel when it is available, and I'll let everybody know that probably on our Facebook page. But the program today is Curtis is gonna talk about the research a little bit. He'll talk about the natural history study a little bit, and then we'll open it up to any questions that anyone has on either of those topics or on GA1 in general. So we're very glad you're all here. We're excited to see all your faces and welcome.
Hi, everyone. My name is Curtis. It's so nice to meet those of you who I've never met before. I see some familiar names and some familiar faces, and so excited to see you again. I am going to go ahead and start sharing some slides, and then I have a couple of people from our team joining me in our office, so I will look over at them to see if they see the slides okay. Great. So I think people are going to be coming in as we get started, but I really am hoping that this is going to be really casual and hopefully meet the things that you guys need. And we would love to open this up to some just informal question and answer, get to know all of you. And hopefully this isn't the first time that all of us talk. But as you may have heard, we're really interested in a relatively short natural history study to support some development of new treatments for patients with GA1. We kind of want to explain why we're doing this and why we think this natural history study is needed at this moment. And I also really want to talk about how excited I am that there are so many new treatments that are in the laboratory for patients with GA1. I've given this talk 10 years ago and sort of felt like new treatments will be coming soon, but didn't have a lot to share. And now we have a lot of really exciting data in mice. And we're hoping that that will move to all of the people who hopefully are on this call or your loved ones. So, first, just a little bit about who we are in case we've never met before. Um, this is our team here in Colorado. You just met Christy, who is our lab manager and the sort of the heart and soul of the lab. I will say Nicole is our current PhD student, and a lot of this work on the natural history study was also done by Courtney here on the screen. Dr. Wootner, who is joining me in the office, and myself, actually both trained with Dr. Steve Goodman. And I don't know if all of you recognize Dr. Goodman's name or not, but Dr. Goodman is the person who first described GA1 in a brother and sister here in Colorado. It's always been fun to listen to him talk about those very first patients with GA1 that he met in the early 70s. And that publication came out in 1975. So quite a long time ago. He really spent his whole career focused on GA1 and GA2, as well as some other metabolic diseases, and did a lot of different things. So, those of you who might have loved ones who are a little bit older with GA1, probably your biochemical testing was done here in Colorado, was really one of the first laboratories to do any of the biochemical testing. Certainly, the only laboratory in the United States who did any enzyme testing. So, if any of you had a skin biopsy, to measure enzyme activity. That was done here by Dr. Goodman's lab. He was really published sort of the first paper on genetic mutations that cause GA1 and was really important, led all of the current treatments and was on those first consensus guidelines. He unfortunately died in 2020, but still is a main reason that our laboratory continues to do this work and we're constantly inspired by him. Also, Dr. Wootner, who is one of our senior scientists, he trained with him. I'm gonna embarrassingly say the year started in the 1990s and helped create the first mouse model of GA1. This is the mouse model that we still, as scientists, use today. And then he joined Dr. Goodman's lab as a fellow in the early 2000s and stayed with him all the way through 2020. So we're Dr. Wootner and I both really talk all the time about the things that we've learned from Dr. Goodman, and we're really excited that we can sort of continue his legacy of GA1 research in our lab. Some of the things that some of you may have recognized me from. I am really proud of the fact that I am even a little bit associated with the Organic Acidaria Association, and we see quite a few members from the OAA here. I definitely saw Kathy on the call. You can see with her nice OAA background, and I think I saw Haley as well come onto the call, and just excited to be just even a small member of this group. I'm also a scientist associated with the Rare Organic Acidemia Research Consortium. This is a very new group study funded by the NIH, where we are focused on all organic acidarias. I'll be honest, my role on this consortium is to constantly say we should do more about GA1. I will say that at every meeting that I ever go to. And there's a lot of organic acidarias, not just GA1, but that's obviously what I'm most interested in. And so we're excited that this just was funded in the fall and we are just starting this 5-year grant. I've also been really lucky to be a scientist on the Charlie Consortium. And this is something that you probably are less familiar with because this is mainly a group of European scientists, and I'm the only US scientist involved. And this group is focused on developing new treatments for patients with lysine disorders, including GA1. And we're going to talk a little bit about some of those new treatments that we've published on our mouse models and some cell lines, but really exciting things that we're hoping to bring to patients. The Charlie Consortium also does a pretty good job of interacting with family groups. So I personally felt really excited that Kathy was willing to come join us here representing the OAA in our first ever meeting in Spain back in 2022. I loved having her there. I hope she enjoyed that meeting. This consortium actually just ended this year. It was run by Clara Van Karnebeek, who is in the middle. I don't think you can see my mouse. But great group of scientists. Again, this whole group of people are focused on developing new treatments. So hopefully you're asking, well, if there's so many new treatments coming out and we can't wait to hear about all of them, why are we still talking about the natural history of GA1? What is the gap that you're worried about, Curtis? And so I'm going to talk a little bit about what we know about GA1 and most importantly, what we do not know. And so I'm going to use some slides that I have used for the last 5 years teaching biochemical geneticists about GA1. So if any of you have a younger biochemical geneticist, they've probably heard this talk. And if they're saying things that you don't think are correct, it's probably my fault. And you should tell me that I need to do a better job training the next group of physicians. So just a little bit about GA1, and this is an audience that knows a lot. I am going to use some numbers from the literature. I am going to talk about things that can happen to kids that we're all trying to avoid. So I want to put that out there. I'm not trying to scare anyone, but as you know, this is still a disease that kids and adults can have symptoms from. The thing that we mainly talk about in clinic with our patients, I'm going to guess the thing you mainly talk about with your doctor, especially when you're young, is this risk of an injury or a metabolic stroke in the brain. And your doctor probably uses different terms. They might call it a striatal injury or a basal ganglia injury or a metabolic stroke. And they all mean the same thing, that there are cells that die here in the basal ganglia. I'm highlighting it with this colored cartoon. And that usually the risk of that happening is the highest when you're very young. And so we know that this stroke has happened in children with GA1, usually shortly after birth, up until the age of 3 years old. And that some kids have had a second stroke. So they've had that first one in those first 3 years of life and have a second one all the way up to 6 years old. We're going to really briefly talk about current dietary treatment and maybe your clinic changes the way that you are on diet at 6 years old. And the reason we use that number is because that is how old patients have been when they've had a second injury. So that's why things change at 6. I'm guessing you've all heard what the basal ganglia is. Some physician teams like to talk about a very specific part in the basal ganglia is injured. And it's usually this part here that is represented in green. So you might hear your physician or your team talk about the caudate or the putamen. But these are all the same parts of the brain. And all of these can result in changes in how we move. And so it can impact on your gross motor skills, like your ability to walk well. It can impact your fine motor skills, so your ability to hold things. It can impact your ability to communicate. And it's very, very different per patient. And we have talked about this as long as we've known about GA1. Oh, I wanted to show one human MRI in case you did not like the cartoons. So this is all these pictures here are from an 11-month-old girl who has GA1 and had some problems with movement. This is all pictures of her brain. And when we are looking for those changes in that basal ganglia, we're looking for these very bright spots here represented by the yellow and white arrows. So it's a very specific part of the brain, and it mainly impacts our movement. Most patients have had this movement change, this injury after an illness. And I'm guessing all of your doctors talk a lot to you about treating fevers and treating when you're sick and if you're throwing up, what to do. Although some of our patients have had this happen without any illness at all, we sometimes refer to this as an insidious onset. That's what it means is we don't have an illness and it still happened. The question that we don't know, and maybe I've talked to some of you about this, or some of you have called me, or your doctors have, is how often this happens. We know that before newborn screening, which is here on the red, somewhere around 90% of every patient with GA1 ended up with that metabolic stroke. So 9 out of 10 patients had a metabolic stroke. We know that that is much better over the last 30 years. Newborn screening started for GA1 in the 1990s. Every state in the United States started doing newborn screen in 2006. And so since we've done newborn screening, and we know that a child has GA1, and we do everything that we can do possible, we've really lowered that risk. But we don't know what it is. Some people say 2% of the time a child can still have a metabolic stroke. Some other people will say high as 30 or 40% of the time a patient can still have a metabolic stroke. And I'll be curious, your doctor might have given you a number somewhere in between. And the truth is, we don't really know what that chance is. And that can be very frustrating to a family. I know the families I've seen in clinic, that's very hard. They want to know an exact risk, an exact number, what is the chance of this. And it can also be very, very hard as we start talking to companies about developing new treatments for patients, as they want to know, are all patients treated very well? If it's only 2%, that means we're doing a very, very good job and we don't need anything new. Or is it a higher number that and we need a new treatment? And that's really one of the things that our natural history study is hoping to find out, both for you so that you have information about your child or your loved one, for your physician team so they have a little better idea of what this risk is. And also as we think about, do we need new treatments for patients with GA1? I think we spend a lot of time as metabolic providers. So if this is not your clinic, that's amazing. But it feels like all we talk about is that metabolic stroke. And we know that kids and adults with GA1 can have other symptoms. They can start to have seizures. In fact, most of the patients who've had seizures start in teenage years. And we don't do a great job of thinking about that risk and how to treat them before they start and when they start. There can be other neurologic findings that happen after 6 years of age. And then like having a stroke or some movement problems that are a little different. A lot of patients tell us that they have headaches or dizziness. And what I don't know, sitting here in front of you, is how likely that is. I can tell there's some of you on this call who have had this conversation with this year and asked, like, what is the chance that my child develops epilepsy? And I don't really know. I don't have that answer. And I would like to be able to say to you, well, in the United States, we've talked to this many families, and it's only 1% of patients, or it's actually a higher number than that. And we've thought a little bit about um, how often you should see a neurologist looking for those symptoms. We know that you can have changes on MRI. Here's a picture of a whole lot of different patients, actually, who all have different MRI findings, but we don't actually know what they mean because we don't have enough data. And then a few adults have some changes with their kidney. And although this is very rare, again, not trying to scare anyone on this call, sometimes people don't have any symptoms till they're adults. This paper came out 2 years ago, and it made my colleagues in metabolism have a lot of conversations about why we're stopping treatment at 6 years. But in this paper, there was a 41-year-old patient who had their very first symptom of epilepsy, and a 51-year-old patient who presented with some significant weakness. And we know somewhere around 9 patients who've been reported in the literature who had their very first symptoms as adults. And what we're trying to do is understand what all of these symptoms are, what all of the risks are, and a bigger question of, are we treating these symptoms correctly? Should we be trying a different approach to keeping you or your loved ones healthy? Um, I wanted to briefly cover a couple of things that people ask us all the time. I'm guessing everyone on this call knows that GA1 is due to a difference in how we make lysine. I think a lot of you probably have been on a lysine-free diet or measure how much protein you eat. And the idea is that as we eat lysine, it gets broken down and used in the body for many different things. It's important in how we make muscle and protein, and it eventually gets broken down. And in patients with GA1, you have this increase of a few different biochemical or metabolites. And your doctor probably talks about these often, your glutaric acid and your 3-hydroxyglutaric acid. We use these to help diagnose patients with GA1. This is how we pick up patients on newborn screening, is that we see these are higher than patients who don't have GA1. And we also think that our treatment should try to lower these. So maybe your doctors measure these while you're on your diet over time. We also sometimes measure enzyme activity. I know that's not overly common, but as I mentioned, Dr. Goodman's lab has been able to measure this for the last 20 or so years. And we can see that most patients' enzyme activity can be a number that is different all over the place. And I think what I want to emphasize here is at this moment, none of these biochemical labs or these enzyme labs really tell us what your chance or your child or your loved one's chance is of developing a metabolic stroke. And that can be incredibly frustrating to you as a patient. It's incredibly frustrating to your doctors. It's frustrating to all of us, is that we can't help figure out what your individual risk is or how to lower that risk. And one way to do that is to collect more information. I want to make it clear that we're not making a promise that at the end of this study or the ROAR study or any of them, we'd be able to tell you that. But this is the first step of, are there things in your blood, in your urine, and your genotype that we can help figure out why you're at more or less risk? People today love genetics. I love genetics too. I've been trained in genetics for a long time. Maybe all of you know your mutation or your loved one's mutation. There's a lot of them. And the point of this slide is to show that there are a lot of them. And so far, knowing that information has also not helped us figure out what your risk is. But we don't put the information together. All of you on this call know a lot about you or your loved one, but we don't put them all together to see if we can really figure out something when we combine data sets. Currently, we tend to treat most of our patients the same way. There's this recommendations for how to treat patients with GA1 that hopefully your clinic follows the guidelines as well as possible. And most of the time, what we are doing is we are making sure that when you're sick, we're giving you a lot of calories, a lot of ways to make energy. And we're also putting you on a diet. Again, we tend to change that diet between whether you're very young or very old. Very old. 6 year. I just said that anyone 6 years old or older is very old, but we do try to change it with age. What I will say that regardless of what that striatal disease is, that metabolic stroke is, something that I think more companies need to hear, we need to hear more voices from families, is that regardless, this treatment is incredibly difficult. And we need better treatments just because this is hard. And even if we developed a new treatment that was as good as the diet, but you could eat whatever you wanted, I would think that would be a huge victory for patients with GA1. We know it's hard. This is a survey that we in Colorado did and published in 2020 of your dietitians. And what we took away from that is that the dietitians thought this was really hard too. And they're not the ones who have to worry about what they're eating. They're not the ones mixing the formula at home. We also saw in this figure that not every dietitian follows the guidelines. So some clinics stop the diet at age 3. It was very little. Most places stop it at age 6, but you might be on this call and still be on the diet at age 10, 12, 13, 20. And hopefully, what we're doing is collecting more data to understand if there is a benefit of continuing this or other therapies past the age of 6, because we know a lot of you are still on the same treatment you were when you were 2 or 3 years old. And again, I really would love to hear more from your voices. This is a nice survey we did. Maybe some of you participated in that Charlie consortium. And we were surprised by some of these numbers that only 30% of families said that their patient, that their loved one or they themselves had no symptoms of GA1, which means 70% of patients around the world thought that they had some symptoms. That the majority of patients were still on some diet even after the age of 6. And that 21% of families who responded to this study felt like they had something that made their symptoms get worse. We asked patients if they'd want a new treatment, and everyone said yes. And then we asked what they'd want a new treatment to really focus on. And a lot of people put in things that involve the brain or the movement, like dystonia or motor problems. But the number of people who just talked about the fact that they would love any treatment that doesn't limit what they could eat, that they wouldn't have to worry about going to a restaurant or going on vacation. And I really appreciated some of the stories the family shared with us. Here's a short one, and I am going to read it. But this was from a family that I got to meet. And they said the responsibility of caring for a child with needs that could have such irreversible and life-changing consequences if not handled correctly has been such a heavy weight to bear. We spent her early months and years petrified of our daughter having a crisis, and we were high vigilance to any minor illness. Hearing their story about how hard it was to simply go on vacation for a weekend and how much thought had to be put into, are we near a hospital? Do we have enough formula? What if we run out of formula? What are our feeding issues? I'm going to say it again that I think we need better treatments for patients with GA1. And I'm guessing no one on this call disagrees with me, but the number of times that we have gone to large pharmaceutical companies or written grants to say, we need better treatments. And the response is, this condition is treated perfectly, is very frustrating. And so one of our biggest goals from this natural history study is to be able to go back to those companies and those funders and say, it's not true. This is not perfect, and we need something better. I want to end on that there's a lot of exciting things that are better. There are amazing researchers around the world who are working on GA1. This Charlie Consortium is a lot of physicians and scientists working together to develop new therapies. What we've mainly done from the Charlie Consortium is work on what is called upstream enzyme inhibition. What we're really trying to do is allow people to eat whatever they want. including as much lysine they want, but stopping that lysine from ever going down this road where it gets broken down and made into these bad or toxic chemicals. And so I'm trying to represent that here with the chemicals turning from red to yellow because we don't completely normalize them. The Charlie Consortium has come up with quite a few number of drugs that are able to do this in the lab. And so are some US scientists. And since we're in the US, I wanted to highlight those individuals who are not part of Charlie Consortium. Here's an amazing scientist and friend at Mount Sinai who has shown that by stopping lysine metabolism from happening in a mouse model, he can make these metabolites normal. Here's another amazing scientist from Duke University who did the same thing. Not only could he make the metabolites normal, but he could keep his mice alive in the mouse model of GA1. When you feed the mice a lot of lysine, the mice die. And they have some very specific changes in the brain. And in this model, he was able to feed them as much lysine as he wanted, and they were able to live. At this moment, we have, as scientists in labs, we have really interesting drugs that have seemed to work that are already FDA approved in animals. We've developed some nice fancy RNA therapies, some CRISPR-based therapies, all the things you might have heard in the news that are treatments for diseases like spinal muscular atrophy and cystic fibrosis. We have all of those drugs in the labs made by scientists. We even have a couple of gene therapy products for those of you who are more excited about gene therapy than other drugs that have completely made cell lines normal again. This is work from the Charlie Consortium where we've just made a cell line from a patient look like a normal cell line. And this is a group from China that has completely cured a mouse model of GA1. So we have all these exciting drugs in the lab from scientists. But what we need are companies who are excited about moving these treatments from our laboratories and from mice to patients. And one of the things that I think is my job as a rare disease researcher, as is your job, and I know Kathy and the Organic Aciduria Association do this very well, is that I constantly email and talk to large companies and I say, there is a need for patients with GA1 to have better treatment. We have exciting things in the lab, and you have an opportunity to make these patients' lives better. And when I get calls back from them and I get meetings from them, they constantly say the same thing to me. We talk to other experts, we read papers, and these patients are treated perfectly. And again, I don't think that that is true. I think that there are still symptoms that patients have that need better treatment. And I think that no one should have to be on such a strict diet the rest of their life. So how are we trying to make a small difference is that we do have a natural history study right now that we are trying to collect that data for all companies, all physicians. This study is actually funded by a large pharmaceutical company named Ann Lyam. So if you enroll in our study, you'll see that in the consent form. That company has started development of a drug for GA1 that they are excited about. I want to make it really clear that no one in my lab or myself own any of these drugs at all. We don't get any money if this drug becomes an FDA approved drug and your child starts to receive it. But they have developed a medication that seems like it works very well in mice. And what we are trying to do is to convince them to keep going. We want them to spend money to have a clinical trial that maybe some of you will be interested in enrolling in. And if that works well in a clinical trial, which we never know, none of you are mice, we want to convince them to keep going past that and try to get this drug to market. And so that is the goal of our natural history study. It is going to be relatively fast because we are trying to convince companies who are on the brink of making a decision about a medication what to do today. We hope that this data, if you enroll, we can share this with all of the other people who are interested in GA1, definitely with our partners at the Organic Aciduria Association who have like a wonderful study moving forward. For those of you who enroll in the ROAR consortium, we'd also like to use that data. We want to partner with everyone, but our goal for this year is really to try to understand the needs of families, the needs of patients, and hopefully convince our partners that there really is a need and an opportunity for them to change some patients' lives. And so that is why we're doing this study. I really want to thank everyone who has been involved in the study from my team. I definitely also want to highlight that this website was made by Sarah Simonson, who I see her on there. Maybe she can wave, and all the partnerships she's done too. And so, with that small discussion, we'd be happy to stop the formal slide part. And I think I see some questions in the chat and happily to talk more about why we're doing this and if you think that this is an important for us to do.